Introduction. Welcome, medical students, to today's lecture on the intricate topic of muscle glycogen metabolism. In this comprehensive script, we will delve into the various enzymes, regulatory factors, and storage diseases associated with glycogen metabolism. We will also explore the dynamic relationship between muscle glycogen and fatty acid oxidation during exercise. So without further ado, let us commence our exploration of this fascinating subject. Glycogen synthase, a key enzyme involved in glycogen metabolism, catalyzes the transfer of glucose units from UDP glucose to an existing glycogen chain. This process occurs in the presence of glycogenin, which initiates glycogen synthesis. The active form of glycogen synthase is composed of two distinctly regulated isoforms, glycogen synthase I, GSI, and glycogen synthase D, GSII. GSI is primarily active in the liver, while GSI is predominantly active in muscle tissues. Phosphorylation is a crucial regulatory mechanism for glycogen synthase. Phosphorylation by protein kinase A, PKA, and glycogen synthase kinase 3, GSK3, inactivates the enzyme, whereas dephosphorylation by protein phosphatase 1, PP1, restores its activity. Hormonal control, coordinated by insulin and glucagon, plays a pivotal role in regulating glycogen synthase activity. Insulin activates PP1, promoting dephosphorylation and glycogen synthesis, while glucagon activates PKA and GSK3, leading to glycogen synthase phosphorylation and inhibition. Glycogen phosphorylase is another essential enzyme involved in glycogen metabolism. It catalyzes the release of glucose 1-phosphate, G1P, from glycogen chains by breaking alpha-1, 4 glycosidic bonds. Glycogen phosphorylase exists in two forms, phosphorylase A, active, and phosphorylase B, inactive, regulated by reversible phosphorylation and allosteric interactions. Phosphorylation of glycogen phosphorylase occurs via the action of phosphorylase kinase, which is activated by phosphorylation by protein kinase A in response to glucagon or epinephrine signaling. On the other hand, synthesis of glycogen phosphorylase is stimulated by insulin signaling and the subsequent activation of protein phosphatase 1, which dephosphorylates and activates glycogen synthase, leading to increased glycogen breakdown. The balance between active phosphorylase A and inactive phosphorylase B is tightly controlled to maintain proper glycogenolysis. Glycogen branching enzyme plays a critical role in glycogen metabolism by introducing alpha-1, 6 glycosidic branches into the growing glycogen chain. This branching allows for efficient glucose release and synthesis during glycogen breakdown and synthesis, respectively. The branching enzyme acts on the nascent alpha-1, 4 glycosidic chains, cleaving a segment and transferring it to another position within the same or adjacent chain. This process generates a branch structure that increases the solubility and compactness of glycogen, facilitating its storage and subsequent utilization. Glycogenin, a unique self-glycosylating protein, plays a crucial role in initiating glycogen synthesis. It acts as the primer for glycogen synthesis by catalyzing the addition of several glucose molecules from UDP glucose to create a glycosylated tyrosine residue within its protein structure. This initial glucose chain acts as the initiation site for further glycogen synthesis, facilitating the growth of glycogen granules. Glycogenin remains covalently attached to the reducing end of the glycogen chain and serves as a primer for future glycogen synthesis cycles. As a result, glycogenin is an essential component for glycogen synthesis and maintenance. Glycogen storage diseases, GSDs, encompass a group of inherited disorders that result from abnormalities in glycogen metabolism. These diseases are characterized by impaired glycogen synthesis or degradation pathways, leading to the accumulation or deficiency of glycogen in various tissues. There are several types of GSDs, each caused by mutations in specific genes encoding enzymes involved in glycogen metabolism, such as glycogen storage diseases type I, GSDI, caused by defects in glucose 6-phosphatase, or GSDV, McArdle disease, caused by a deficiency in muscle glycogen phosphorylase. These diseases manifest with diverse clinical symptoms, including hepatomegaly, muscle weakness, hypoglycemia, and exercise intolerance. AMP-activated protein kinase, AMPK, is a master regulator of cellular energy homeostasis, including muscle glycogen metabolism. It exerts its regulatory effects by sensing the AMP/ATP ratio, which rises during energy-requiring activities, such as exercise. Upon activation, AMPK phosphorylates and inhibits glycogen synthase, promoting glycogen breakdown and inhibiting glycogen synthesis. Concurrently, AMPK activates glycogen phosphorylase, leading to increased glycogenolysis and glucose release from glycogen stores. These actions collectively enhance the availability of energy substrates required during times of higher energy demands, ensuring the maintenance of adequate cellular energy balance. Hormonal regulation plays a significant role in governing muscle glycogen metabolism. 3 hormones, insulin, glucagon, and epinephrine, exert distinct effects on glycogen synthesis and breakdown to maintain glucose homeostasis. Insulin, released in response to increased blood glucose levels, activates protein phosphatase 1, PP1, which leads to dephosphorylation and activation of glycogen synthase. This stimulates glycogen synthesis and storage in muscle tissues. On the other hand, glucagon, released during hypoglycemic conditions, activates protein kinase A, PKA, resulting in phosphorylation and inhibition of glycogen synthase. This promotes glycogen breakdown and glucose release from glycogen stores. Epinephrine, a stress hormone, acts through alpha and beta adrenergic receptors to activate protein kinase A and phosphorylase kinase, leading to increased glycogenolysis and subsequent glucose release in response to fight or flight situations. Glycogen degradation, known as glycogenolysis, involves a series of enzymatic steps to convert glycogen back into glucose 1-phosphate for energy production. The enzymes involved include phosphorylase, phosphoglucomutase, and glucose 6-phosphatase. Initially, glycogen phosphorylase cleaves off glucose 1-phosphate from the glycogen chain by breaking alpha-1,4 glycosidic bonds. Subsequently, phosphoglucomutase converts glucose 1-phosphate to glucose 6-phosphate, which enters glycolysis or gluconeogenesis pathways. In the liver, glucose 6-phosphatase further converts glucose 6-phosphate to glucose, which is then released into the bloodstream. Glycogen synthesis is a highly regulated process involving several enzymes and substrates. The synthesis pathway primarily occurs in the liver and muscle tissues. UDP glucose plays a vital role as the substrate for glycogen synthesis. It is generated from glucose 6-phosphate by the action of UDP glucose pyrophosphorylase. Subsequently, glycogenin facilitates the attachment of UDP glucose molecules to create the primer for further glycogen synthesis. Glycogen synthase is the key enzyme responsible for elongating the glycogen chains using UDP glucose as the glucose donor. At the branching points, glycogen branching enzyme introduces alpha-1,6 glycosidic linkages, enhancing the compactness and solubility of the glycogen molecule. Glycogenolysis regulation involves intricate interplay among various enzymes and hormonal signaling. Hormones such as glucagon and epinephrine stimulate glycogen breakdown through their respective signaling pathways. Glucagon activates protein kinase A, PKA, which phosphorylates and activates phosphorylase kinase, leading to the conversion of phosphorylase B to phosphorylase A. In turn, phosphorylase A catalyzes the release of glucose 1-phosphate from glycogen, resulting in increased glycogen breakdown. Epinephrine, in addition to activating PKA, also activates glycogen phosphorylase kinase, enhancing its activity and promoting glycogenolysis. The synergistic effects of glucagon and epinephrine ensure the coordinated mobilization of glycogen stores during energy-demanding situations. Exercise exerts profound effects on muscle glycogen metabolism, impacting both glycogen utilization and subsequent recovery. During exercise, muscle contraction stimulates calcium release, activating glycogen phosphorylase and promoting glycogenolysis. As exercise duration increases, glycogen stores become depleted, and reliance on other energy sources, such as fatty acid oxidation, becomes more prominent. Upon cessation of exercise, glycogen synthesis pathways are facilitated by increased insulin sensitivity, allowing for replenishment and restoration of glycogen stores. Muscle glycogen metabolism intertwines with fatty acid oxidation during exercise to maintain energy supply to working muscles. As glycogen stores become depleted, fatty acids derived from adipose tissue triglycerides are mobilized and transported to muscle cells. The breakdown of fatty acids into acetyl-CoA molecules occurs through beta-oxidation within muscle mitochondria. Acetyl-CoA then enters the Krebs cycle, yielding energy-rich molecules, such as ATP, to fuel muscle contractions. Simultaneously, the oxidation of fatty acids helps to spare limited glycogen reserves, ensuring their preservation for bursts of intense energy demand. The balanced interplay between glycogen metabolism and fatty acid oxidation is critical for optimal exercise performance and endurance. In conclusion, muscle glycogen metabolism is a highly regulated process involving various enzymes, hormonal signals, and feedback mechanisms. Understanding the intricacies of glycogen synthesis and breakdown is essential for comprehending the pathophysiology of glycogen storage diseases, GSDs. The interplay between hormonal regulation, such as insulin, glucagon, and epinephrine, governs the balance between glycogen synthesis and breakdown to maintain glucose homeostasis. Furthermore, mechanisms involving AMP-activated protein kinase, AMPK, and the relationship between muscle glycogen metabolism and fatty acid oxidation during exercise contribute to overall cellular energy balance. As medical professionals, comprehending the nuances of muscle glycogen metabolism equips us with the knowledge to diagnose and manage glycogen storage diseases effectively. It also provides a foundation for understanding the intricate metabolic adaptations that occur during exercise. Thank you for your attention, and I hope this detailed script has provided you with a comprehensive understanding of muscle glycogen metabolism.